So the first task, I'll give you a little bit of time to go through this, but it's really important that you can write the equation for photosynthesis, not to be confused with the equation for respiration. They are very similar, but in reverse. For combine, can you think of any factors that would limit the rate of photosynthesis? So can you think of anything that may make photosynthesis happen faster or slower if it was there in greater or fewer amounts? And for create, this is thinking more along a practical kind of idea. How would you be able to test whether the rate of photosynthesis had sped up or slowed down? So how many of these did you get? If you got all three of these, well done. A common mistake that students often do is to include water on this list. So if you wrote down water, well done for considering it, but it is actually not considered to be a limiting factor most of the time. This is because most of the water that the plant takes in is used for other purposes, such as support, keeping the plant upright, and for transportation of substances through the plant. It's only used in relatively small quantities for photosynthesis, and a plant would end up suffering from lack of water and wilting and going on to cell death before the lack of water had an effect on photosynthesis. So this is the reason why it's not considered to be a limiting factor, even though it is one of the reactants in the equation. So this is a practical which is common at this point in the topic, and in fact, had we been in school, this would have been a core practical, a required practical that you would have completed yourselves. So in the diagram on the left, you can see there's a lamp, there's a meter ruler to measure distance, and then a beaker full of water and a boiling tube with some pondweed in it. As you can see, there are oxygen bubbles being produced by the pondweed coming out through the water in the boiling tube. On the right hand side, there is a table of data. So there is distance from the plant in centimeters, and then units of light intensity and number of bubbles per minute. In this practical, you would measure the distance from the plant of the lamp and also the number of bubbles that you could count in the minute. And you would then, as highlighted with the red shape, calculate the intensity of light using a mathematical formula, which you will do if you try the extension sheet that is also included in today's lesson. If you want to try and produce a graph to improve your IT skills, then there are instructions on the Google Sheet that I've shared so that you can create a graph of your own which looks similar to the ones that you'll use later on in this presentation. As part of this practical, there are some considerations that you would need to consider, and one of them is why you might place the pondweed inside a beaker of water in front of the lamp. Why not just place the pondweed inside the boiling tube and then place that in front of the lamp? So think about what advantage there's going to be of having the beaker of water there. If you have a look at the diagram, it explains this. That's something that you might be asked in a question that came up on this topic. So a key skill in this topic is to be able to analyze graphs. What this basically means is you are describing and explaining what is going on the graphs. and it is the quality of your language and the detail that you include, which is what will make the difference between you getting good marks or losing marks. So on this graph, this is a typical graph that you would get for a rate of photosynthesis experiment, and this will be similar to the graph that you produced on the Google Sheet if you had to go at that one. And you'll see that there are two different phases, and they're identified on the graph as 1 and 2. Now, although there aren't any numbers on the graph, you can still talk about these factors changing in terms of increases and decreases, or remaining constant. And if you have a look at the x-axis for light intensity as you go towards the right, in the direction of the arrow, this is light intensity increasing towards the right. So, phase 1. We're going to describe phase 1 up to the point in the graph which is highlighted by the red circle. So the description is coming up, and the description would be, as the light intensity increases, the rate of photosynthesis increases. So the line is traveling upwards. As light intensity is increasing, as we move towards the right of the graph, the rate of photosynthesis is also increasing as we go up the graph. Now we need to get on to the explanation part of the analysis. So this is where you need to refer back to how light is involved in the photosynthetic equation, and the function of light is to provide the energy needed for photosynthesis. So our explanation would be, as illustrated in the box, light is needed to provide the energy for photosynthesis. So as light intensity increases, more light can be absorbed by the chloroplasts and photosynthesis occurs faster. You'll notice in that answer there is quite a lot of detail. So we've referred back to what we said in the description, as light intensity increases, and we've also included some extra information about what parts of the plant and the cell are involved in photosynthesis relating to the chloroplasts and how they are involved in the process, so they are absorbing the light. And we've also related to the fact that photosynthesis can therefore occur faster. So there's quite a lot of detail in there to get the marks, and this is something that you will practice over the course of the next couple of slides. Moving on to phase 2, we now see as light intensity increases, or as we go towards the right of the graph, the rate of photosynthesis doesn't go up anymore. The words we use for this is that it remains constant. You can say stay the same, and you probably would get the mark, but try and get used to using the language that is the level of detail that you need. So going through the description, as the light intensity increases, the rate of photosynthesis remains constant. Try and use this pattern of language, starting with the independent variable on the x-axis, the light intensity, and then referring to the change in the y variable, the dependent variable, rate of photosynthesis. And talk about it in terms of as the light intensity increases. Don't talk about change. Change doesn't mean enough when we come to GCSE science. You need to talk about what that change actually is. The explanation for that is going back to the idea of there being limiting factors and more than one. So start off by saying that light is no longer the limiting factor, and you can use the same pattern of language for when you come to the other graphs later. Then there must be another limiting factor which is preventing photosynthesis from occurring any faster, and almost always you will get a mark for stating what this might be. So for example, temperature or carbon dioxide concentration. Remember, water is not, so don't include that in your answer. Okay, over to you. So here you see there is a different graph. It looks almost identical. It has two phases, but you'll notice on the x-axis we've got percentage level of carbon dioxide now, as opposed to light intensity. You can use exactly the same pattern of language as I gave you on the previous slide to answer this one, to describe phases 1 and phase 2. Just be careful of the variables that you're talking about and get those inserted into the correct places, but otherwise the pattern is still the same. So here we have the final type of graph that you would get when talking about the limiting factors on photosynthesis, and this is for temperature. And here you have also been given a value of 40 degrees, which is placed around about in the center, and this is towards where the number 2 part of the phase is highlighted on the graph. So this graph has 3 phases that are important to discuss. Number 1, as the rate is increasing, number 2, where it reaches a peak, and then number 3, when it decreases. And for this answer, when you're talking about what's happening, it's important that you refer back to your understanding of what happens to some reactions above a certain temperature, linked to another common idea in biology. If you want a hint about this, there'll be a hint on the final slide of the presentation. But use the same pattern of language to describe and then explain what is going on at each of the phases on this graph.